Glutamate is the principal excitatory neurotransmitter in the central nervous system and plays a pivotal role in synaptic plasticity, the cellular mechanism underlying learning and memory. Synaptic plasticity refers to the ability of synapses, the connections between neurons, to strengthen or weaken over time in response to patterns of activity. When an action potential reaches the presynaptic terminal, glutamate is released into the synaptic cleft and binds to postsynaptic receptors, primarily AMPA and NMDA receptors. The activation of AMPA receptors leads to a rapid influx of sodium ions, causing depolarization. If the stimulation is strong or repetitive, NMDA receptors are also activated, allowing calcium ions to enter the neuron. This calcium influx is a critical trigger for intracellular signaling cascades that modify the strength of the synapse. Long-term potentiation, LTP, is a well-studied form of synaptic strengthening and is considered a key process in memory formation. During LTP, repeated or high-frequency stimulation of synapses results in sustained increases in synaptic efficacy. Glutamate-mediated activation of NMDA receptors initiates molecular events that increase the number and sensitivity of AMPA receptors at the synapse, enhancing the postsynaptic response to future glutamate release. Conversely, long-term depression, LTD, is a process of synaptic weakening, often induced by low-frequency stimulation. In LTD, glutamate signaling through NMDA receptors leads to a modest influx of calcium, activating pathways that result in the removal or deactivation of AMPA receptors from the synapse. This decreases synaptic strength, allowing for the fine-tuning of neural circuits and the elimination of unnecessary connections. Through these bidirectional mechanisms, LTP and LTD, glutamate signaling dynamically regulates synaptic strength, enabling the brain to encode, store, and modify information. Disruptions in glutamate signaling or receptor function are associated with cognitive deficits and neurodegenerative diseases, highlighting the importance of glutamate-mediated synaptic plasticity in learning and memory.